today's video I'll be mainly focusing upon the most important, very important question, what is very frequently asked in the professional exams for microbiology. So where do we see what is culture media and the classification of culture media. So in this video I'll be mainly focusing upon this. So culture media, this is also called as growth media, and in microbiology this has been used, and it is defined as a bacteriological media used for isolation and characterization of bacterial pathogens. So whenever in microbiology, in pathology, any kind of a pathogen is being confirmed as to what is the causative organism. So basically in sputum culture and different swab cultures in different secretions, this culture medias are being very frequently used for the characterization of bacterial pathogens, and they are also given another name. They are also called as growth media. All right, so they have been also given another name which is called as growth media, and in this video we'll be studying about the classification of this culture media. So based on the physical nature, the culture media can be broadly subdivided into three types. They can be liquid media, they can be solid media, or they can be semi-solid media based on the oxygen tension. So if it is an oxygen tension, oxygen is present or absent. So on this it is aerobic media or an anaerobic media. Based on the functions, they can be either simple or basal media. For example, the best example is nutrient broth. The next is complex media, and the last is the synthetic or defined media, which includes the examples is the simple peptone water media. And last we have the semi-defined media. So there are four types in based on the functions. Okay, and there are certain special media. So these special media can be the special media, they are further classified as into the following, such as enriched media, example blood agar and chocolate agar, and egg media, they fall under the enriched media. The enrichment media, it is the tetrathionate broth. The selective media, there are certain bacteria which grow in a particular media, so for that selective media is needed. So selective media, the exact examples is deoxycholate citrate medium for dysentery bacilli. And next we have differential media, where a particular color differentiation will occur. So differential media, the best example is McConkey's medium. And the last in this category of this special media, there is something called as the indicator media. So example is incorporation of sulfite in Wilson's and Blair medium. So this is the example of indicator media. And the last what falls in the special media, it is the transport media, which is the Stewart's media. Now based on the uses, the culture media, they can be classified into medium used for the isolation and growth. The next is medium used for the biochemical tests, and the media what are used for transport. So this was in a very precise, rough manner, I have summarized in a very broad perspective that what are culture media. So these are used for the characterization and isolation of bacterial pathogens. They can be liquid, semi-solid, or solid, and they, based on their uses, they can be classified into the media what is used for isolation and growth, the media what is used for biochemical tests, and lastly the media used for transport. So students, if you like this video, give this video a thumbs up. If there are certain topics from microbiology you want it to be discussed by me in English or in Hindi, you are most welcome to comment me in the comment section. Thank you for watching.